In this section, we're going to talk about gene cloning, and we're going to talk about how we can use this to improve human health. And I like to tell the story of when my kid was diagnosed with type 1 diabetes. So 2 days, 3 days after they turned 16, and 1 day after I turned 40, my kid said, I'm not feeling well. And I said, I guess we'll go to urgent care. And they said, you get to go to the hospital after they took her blood sugar. And I was like, I don't think I want to do that. And they were like, I don't care what you think. And so then we went to the hospital and we found out that this kid's pancreas can no longer make the protein hormone insulin. So most of us, when our blood sugar reaches a certain level, that triggers the production of insulin, which then will allow the sugars to enter our cells, and we can do regular cell respiration and all the things that we've been talking about in this class. This kiddo can't do that anymore because they have an autoimmune disease. So in order for this kid to be able to get energy from her food and also to not get really, really sick from having too high of blood sugar, she needs to take insulin regularly. And the way that that insulin is produced is through this process of gene cloning that we're about to walk through. Okay, I know this is a lot on this slide, but we're going to walk through it. We're going to break it down into each bits. So molecular cloning or gene cloning is how new DNA molecules are going to be produced by combining two different chunks of DNA. And so we need to remember some terminology and introduce some other new terms. Okay, so we've talked about plasmids before briefly. Plasmids are these small circular molecules of DNA and that are found a lot in bacteria, some in eukaryotes, mostly in bacteria. And these plasmids are going to replicate on their own, independent of what the chromosomes in the nucleus are doing or in the nucleoid region are doing. Okay. We're going to take one of those small circular plasmid bits of DNA and we're going to introduce a new chunk of DNA that we want to have that gene be expressed. So this is going to be our foreign DNA. It's not found naturally in that bacterium. Okay. Our host DNA is the DNA that is usually found in that bacterium. So it could be this plasmid or it could be the chromosomal DNA of the bacterium. Okay. So let's walk through what's going on in this image here. Okay. So here's our little small circular piece of DNA. This is our plasmid. Okay. What we need to do is cut that open using these specific enzymes. And there's a couple of ways we can do it, but if you were taking my Bio 181 class, I would make you remember those, but we're not, so we're just going to open up this circle, okay? That's all we're doing. And we're going to do, we need to do that so that we can take this gene of our interest. So for example, the gene that produces human insulin, and we're going to take that foreign piece of DNA and we're going to shove it in that plasmid. Blink. Okay. And then what we need to do is then convince bacteria that they want this plasmid. This is valuable DNA. And so we can actually chemically induce the bacterial cells to chomp up this DNA plasmid. Okay. And then what we're going to do is have them go ahead and produce that gene that we're trying to get. Okay. So again, these restriction enzymes, these are enzymes that are going to cut the DNA at a very specific sequence, right? And so we can have them be cut with these blunt ends, or we can have these sticky ends where those are fragments that are hanging over. We don't need to memorize that for this class. Okay. But that foreign piece of DNA is going to match up with these pieces of DNA that are hanging over. Okay. And then that's how we're going to attach that foreign DNA into this host plasmid. And once we do that, we call this a chimeric molecule. So if you are a fantasy nerd like me, you might know about chimeras, which are a bunch of these, have these different parts of all these different organisms, and they're kind of shoved together to create this new monster that doesn't actually exist in the world. So our recombinant DNA is that plasmid plus, so our plasmid in this image is this yellow bit, and the foreign piece of DNA is this blue bit. This recombinant DNA does not exist in nature, but we can make it exist in a lab. In order for a plasmid to be useful for this process, we need to have three things present. We need to have an origin of replication. That means that's a starting point for the expression of those genes. Remember, we talked about transcription and translation last time. We usually want this inserted gene, right, this foreign piece of DNA, to be expressed. We usually want whatever the product is. We want that protein. So in order for that protein to be produced, we need to have it be the gene, be transcribed and translated. Okay, so that origin of replication is going to be start here, and we're going to be able to go that way. Okay. The restriction site is also called the multiple cloning site. This is where those restriction enzymes are going to come in as a pair of scissors and cut that DNA so that we can pop in that gene. Okay, so far this all makes sense. Yep, we need a starting point. We need a place to cut and paste. But the third thing is a little bit curious. We need antibiotic resistance gene. So without this gene, we need to have, it's not going to really work. We need an antibiotic resistance gene. Why? Well, we need a couple reasons. First of all, if you're a bacterium, you need to want this plasmid. If you don't have this plasmid, you're going to die, right? So having this antibiotic resistant gene gives you an evolutionary advantage, right? You're going to be able to live in new environments, in more environments where there's fewer competitors. So if you're growing where there's antibiotics, if you have this antibiotic resistant gene, you're good to go, bro. You can keep living. The other reason why is because we want all the bacteria cells that don't have this gene to die, and the bacteria cells that do have that gene to live, so that we can harvest whatever this gene product is.